Misha's job here at the castle was to study the chemistry of white blood cells. He decided to look at the large nucleus at the centre of the cell and find out what it was made of. To do this, he needed two things: a ready supply of cells and a way of getting rid of the gloop so he could study the bare nucleus. Getting hold of white blood cells would normally have been tricky, but Tübingen was the ideal place. The region had been at war with Prussia. Hundreds of injured soldiers with infected wounds lay in the barracks next to the hospital. Their wounds were oozing copious amounts of pus, which is full of white blood cells. So, revolting as it sounds, Misha collected their old bandages so he could scrape off the pus. Misha needed something else. His next stop was the local slaughterhouse to collect a pig's stomach.
Oh, look, here it is.
Oh, it's disgusting. He was interested in the mucus that lined the stomach. This contains an enzyme called pepsin, which helps break down and digest food. Where's the pepsin?
Look here, this is the pepsin.
Ah, this sort of gloopy stuff.
Yeah.
There's not much of it.
No, that's all.
And this is what helps digest the food in the stomach?
Yeah.
Smells like a pig's stomach to me. With the pepsin, Misha now had all he needed for his experiment. And this is a delicacy in Tübingen?
Yes, you can fill it with bread, or you can cut it in stripes and then you cook it and eat it with bread.
Is it tasty?
Yes.
I'm not sure about that. Misha carried the bandages and the pig's stomach back to the lab. If he was right, the pepsin would break down the white blood cells. Then, for the very first time, it would be possible to examine the dense nucleus at the heart of the cell. Nowadays, we do analysis like this using precision equipment. But of course, Misha didn't have any kit like that, so it wasn't easy. First, he had to scrape the pus from the bandages. Now, this is mayonnaise, but you get the idea. Then, he had to wash the pepsin out of the pig's stomach using an acid and mixed it with the pus. After the enzyme had done its work and digested the cells, only then could he analyse the nucleus on its own. Now, the big question was, what did the nucleus consist of? Misha spent months analysing its chemistry, and he found it contained a rather strange molecule. This molecule was made up of carbon, hydrogen, oxygen, and nitrogen. He knew these elements were found in all living things. But this molecule contained something extra, phosphorus, and that made it different. Very different. It was an entirely new kind of molecule. Because he found it in the nucleus, Misha called it nuclein. We now know it as DNA. Intrigued, Misha repeated his experiment on sperm cells from frogs, carp, bulls, and salmon. Every time he found exactly the same molecule. Incredibly, some of it has survived. Here in this test tube is some of the first DNA ever isolated. It's DNA from salmon sperm that Misha extracted in the 1870s. Now, it may not look like much, but this brown powder marks the beginning of a scientific revolution.